Yes.
Jai Hind, good evening, and welcome back to all the respected dignitaries, distinguished speakers, faculty members, researchers, students, and participants joining us from different parts of the countries and across the globe. It gives me immense pleasure to welcome you all to this 10 days international training come workshop on advances and innovation in veterinary science for viable livestock practices. This international training program is being organized by College of Veterinary Science and Animal Husbandry, Jalukie, Nagaland, under the esteemed Vital Biotech Group and AFTFS. This program is being conducted under the visionary leadership of our partner, Peren Dr. Anupam Mishra, Honorable Vice Chancellor, Central Agriculture University, Imphal, Chief Patron, Professor Indra, Director of Instruction, CAU Imphal, Chairperson, Professor Lalita, Director of Research, CAU Imphal, Convener, Dr. I. Shakuntala, Dean, College of Veterinary Science and Animal Husbandry, Dr. Amrit Gogoi, convener, Dr. Dinesh Kumar, the course coordinator, and Dr. Jitendra Mehta, President AFTEFS, along with the dedicated efforts of organizing committee and faculty members of College of Veterinary Science and Animal Husbandry, Jalukie. This 10 days international training come workshop is being conducted on an online platform, Zoom, bringing together the experts and participants to share knowledge and innovation in veterinary science and livestock practices. I, Dr. Nisha Rana, warmly welcome you all. So, now moving forward to the technical session 1 for day 8, I would like to introduce our first speaker of day 8. So, our first speaker is Dr. Supan Kumar Mehti. He is serving as professor and head of the Department of Veterinary Surgery at the College of Veterinary Science and Animal Husbandry CAU, Jaluki, Nagaland since 15 March 2024. Prior to this, he worked as principal scientist at IVRI Izzatnagar. He obtained his master in veterinary science and PhD in veterinary surgery from IVRI Izzatnagar. With over 30 years of professional experience, he has successfully guided 11 PhD scholars and 9 master's students and has authored more than 350 publications in national and international journals. Now, I would like to request to Dr. Supan Kumar Mehti to kindly deliver his keynote lecture for session 1. Dr. Mehti.
Hello. Good evening.
Good
Can you
evening,
hear
sir.
me?
Yes, sir, you are audible.
Okay. Okay. Uh, myself already Dr. Nisa introduced me. Myself Professor S.K. Maiti. Actually, last more than 30 years I was at Indian Veterinary Research Institute Surgery Department as a principal scientist. On 24th 15 March on 2024, I joined here in this college, Central University Nagaland Veterinary College, as a professor head and surgery. Now, uh, actually, my my passionate, you can say my passion in some aspects of surgical affections like stem cell research. More than last 20 years, I have devoted in stem cell research. So in this deliberation, I'll not deliver it nothing new. Only I can share my experience, and all the slides, all I will show you. It is not from any other net or any other book or publication. It is my own research. Okay, so I next slide, please. Next slide, please. Okay, next slide. Okay, now I do not know how much interest about this my topics, but I'll try my best to make interest to you all. Now, stem cells. Now, stem cells, as per Nature publications, they define the stem cells as a miracle cells or wandering cells. Why they call miracle or wandering? Because this cell nowadays hue and cry in all over the world, all over the research lab. For as these stem cells can treat so many non-treatable diseases in human as well as the animal. Say, for example, Parkinson disease, paralysis. Okay. Now HIV also, HIV, so many things nowadays, and cancer. Last but not the least, cancer. Now, all scientists from all over the world, they are trying to treat this dreaded disease in our mankind by using stem cells. So, I'll first I will take some few minutes. What about stem cells? What is the definition? From where you get, how you culture it, how you propagated it. Then I'll give in some my experiment in different affections. Say for bone healing affections, skin wound healing affections, in burn wound affections, corneal injury affections, then in liver cirrhosis or liver damage person, paralysis person, and last on cartilage, which never regenerate cartilage nerve. But I'll show you how stem cells help for regenerations of nerve as well as the cartilage. Okay, so what is stem cells? Actually, stem cells मैं थोड़ा हिंदी बोल रहा हूँ. स्टेम सेल है कच्ची मिट्टी. ओके, कच्ची मिट्टी. यू कैन दिस कच्ची मिट्टी को आप so many idols, so many devils, so many idols, so many humans, so many pets. You can make as you like. Kachi mitti ko. This stem cell just like this. It is the self-renewing divisions, and these cells can differentiate it what not. All type of cells, nerve cells, bone cells, cartilage cells, muscle cells, heart cells, all type of cells wherever needed in our body. That is the unique property of the stem cells. So I go hurriedly about the scientific definition of stem cells. Stem cells undergo self-renewing cell divisions. That is, they can give rise to at least one daughter cell that is identical to the initial cell. A character is required to maintain the stem cell pool. Stem cell undergo lineage commitment and differentiate, giving rise to more undifferentiated progenitor cells. Stem cells repopulate in a robust fashion in giving tissue in vivo. Next slide, please. Next, in this diagram, you can easily in middle that is we can develop stem cell from the adipose tissue, or that is called fat derived stem cells. These stem cells can be different, it is multipotent cells, and can be different into the muscles, liver, fat. Okay, then gene therapy can be used in cartilage therapy, fracture healing, paralysis, nerve injury, and in the heart injury. So, in a nutshell, stem cells can differentiate all type of important cells needed in our body in different healthy conditions as well as in the disease condition. Next slide, please. Next slide. Okay. Now, there is an international society that is called International Society of Cell Therapy, ISCT. They have given three criteria. Just if one cell they fulfill these three criteria, then you can only call this is stem cells. First criteria, it should be plastic adherent in standard culture conditions. Either say T75 plus or 225. It is plastic adherent. You cannot may not see when you wash it by the DPBS. You cannot see these animals, see these cells. But when you go to the microscope, these cells you can find it is attached, adherent with the plastics. Second, it must express some positive, say for example, CD45, CD44, CD73, CD90, CD105, as well as some negative surface like CD14, CD34, or CD45. And third, it must differentiate at least either bone cells, that is osteoblast, or fat cells, adipocytes, and cartilage cells, chondroblast. So these three criteria, each any cells fulfill either plastic adherent, they must express some positive as well negative surface markers, and they differentiate at least 3 to 4 different type of cells. Okay, then you call it is a stem cells. Now, actually, these stem cells or mesenchymal stem cells, it is present in our body, but the quantity is very, very less. I just give you an example. In bone marrow, it is 1 into 10 to the power 5 cells. Adipose tissue, 2 in 100 cells. In cord blood, 0 to 2 in 500 cord blood cells. And Wharton cell, 1 in 333 number of cells. So, in this references, I giving some idea that stem cells already present our bone marrow or adipose tissue or umbilical cord blood or Wharton cell, but their number is very, very less. So for therapeutic purpose, you need lots of cells. So you have to isolate the cells. You have to culture passage 1, passage 2, passage 3. After passage 3, you will lot of million cells. Then you can inject or introduce in your body for different type of affections. Next slide, please. So I already told mesenchymal are multipotent cells and they differentiate into different type of body cells. Now the sources, they can already nowadays lot of source. Every day they are researching lot of source, but generally they from umbilical cord blood, chorionic villi of the placenta, Wharton's jelly, liver, lungs, fat, periosteum, teeth, amniotic fluid, symbiosis, and circulatory systems. In my experience, bone marrow is the one the richest source, okay, particularly the epiphyseal, not the diaphyseal. In epiphyseal pen, where the red bone marrow presents, this stem cells is a optimum number of cells you can get from the red bone marrow. Okay. Now, next slide, please. Now, why stem cells are so important? Why whole world it is called wandering cells or miracle cells? Because they have 6 to 7 unique properties. Unique properties. That's why they are called wandering cells. First, they release some trophic support, trophic cytokines, say for platelet derived growth factor, transforming growth factor beta, okay, bone morphogenic protein, interleukin organ 2, so many, and BDGF, so many trophic cytokines they produces, and all have some distinct usefulness in our body. Okay. Second is called homing. One peculiar characteristic is called homing or chemotaxis. Now, if you are fracture healing in any which bone, say for radius ulna or forelimb or hind limb, anywhere, if you inject the MSC, these cells directly go the fracture site. If there is liver damage, if you inject MSC, it is directly go to the liver. If there is any nerve injury, these stem cells after injection, it will go to the where the nerve is injured. So it is unique properties called homing. Third is called revascularizations. After injury, there is some vascularity problem, but MSC helps in revascularizations. Okay. Then it is, I told, differentiation. This MSC can differentiate all type of body tissue, all type of body tissue. And then anti-apoptosis. Now every cells have certain lifespan, but MSC they release some cytokines. It prevent apoptosis, means program cell death. So it is having anti-apoptotic effect. And last but not the least, very important properties of MSC called immunomodulations. That means they are immune privilege. They are immune privilege. They do MSC does not express MSC2 class antigens, and they also suppress T cell proliferation. That's why now human, for human, any liver damage or human, any bone fracture, you can inject rabbit stem cells. You can inject rat stem cells. I'll show you in my rabbit experiment. I use dog MSC, I use goat MSC, sheep MSC, guinea pig MSC. There is no immunogenic reactions. That is the unique properties of MSC. Okay, next slide, please. Now, next slide, please. Okay, I will hurriedly go how you isolate and cultures. Okay, actually, I told you bone marrow is the rich source of stem cells, particularly the epiphyseal part of bone marrow or crest of ileum. And in case of pig, you can use from sternum or epiphysis. Okay, you take a sterile bone biopsy needles. Okay, just you rinsed with heparin and you put in the crest of ileum directly, and by the negative pressures you just withdraw the stylet and you will find the bone marrow is coming. Okay, after say for example you on one part of crest of ileum 2.5 ml, another side, left side or right side, another 2.5 ml bone marrow, total 5 ml bone marrow. Okay, you would get to 5 ml bone marrow. Now you have to prepare one complete media, stem cell culture media. Generally, we put 85 ml of low glucose DMM, the dalbucal modified Eagles medium, and 15 to 20% ABS, just 1% antibiotic, that is penicillin or plus streptomycin or amphotericins when there is a chance of fungus infections. Okay, so you have prepared this complete media. After taking the bone marrow from the animal or from the human, okay, in a 15 ml, 15 only in a 15 ml glass vials, first you put 5 ml fecal plaque or histoplaque. After that, you just slightly dissented from the wall of the pipe, or wall of the glass vials. Okay, and after that, not mix it, not just there will be layer. Below there is fecal plaque and above there is bone marrow with the complete media. Then you go for centrifuge, 3000 rpm 30 minutes, and you will find in interface there will be the stem cells. I'll go directly go there. Okay. Okay, I just show you. Sir, next slide please, sir. Next slide. Sir, next slide. Next slide. Sir, next slide. Sir, next slide, please. Next time. Next
Already
slide,
done,
please.
sir. Already done.
No. Next, I'm going next. Next.
Okay, okay.
Oh, this. I just go collection of bone marrow. Okay. I just go. You just see from the small animal, say for rabbit, I from the crest of ileum. In the dog, below you from the crest of ileum. Next slide, please. From the sheep, also crest of ileum. From the pig, as because crest of ileum, lots of fat, I prefer in the sternum. Okay, directly sternum. But you have to care, you not go through a sternum directly, then you puncture also heart. So sternum, from the sternum you can collect the bone marrow. From the goat, crest of ileum, you can find my slide. I have already taken about 5 ml of bone marrow. Okay, next slide, please. Then already I told you just first taken 5 ml of fecal plaque or histoplaque. Okay, and then layering this bone marrow with the complete media. Then you go for centrifuge. You will be fine. After centrifuge, below there is RBC, next media, and above there is plasma. Okay, above there is plasma. You just see in between plasma and media there is a white transparent. That is called interface. Only that is buffy coat. This buffy coat only present the stem cells. So you discard the above plasma and rest these are either RBC and media. You just collect the interface or Buffy code. These Buffy code are rich in stem cells. Next slide, please. Next slide. Next slide, please, sir. Okay. Then this Buffy code, you take it. Okay. You take next and you go for culture. First P1 says you just put about 10 ml of complete culture media and 1 to 2 ml of Buffy code, about 4 to 5 first you put to T25 flask and keep in a 5% CO2 incubator. Okay, and 4 to 5 days you just started. You show the development of the MSC. Okay, so no, first slide, sir. Rabbit, उसका पहले first canine से पहले. You just see that is called a rabbit MSC. Okay, this MSC you just see spindle shaped. All MSC like spindle shaped. They have a nucleus and cytoplasmic process. These nucleus having more than 2 to 3 nucleolus. Okay, sir. This is so this is a rabbit MSC. Next slide, please. This is canine or dog MSC. Next slide, please. Sheep or ovine MSC. Then rat MSC. Next slide. Okay. Next slide. That is pig BMSC, bone marrow MSC, or it is called BMSC. You can see. Next, that is goat BMSC. Next slide, please. Next slide. That is equine or horse stem cells. Okay. Okay. Actually, this equine I take either bone marrow and from umbilical cord. Okay. Above 2 is from the bone marrow and below 2 is the umbilical cord. So when at first you for P1 passage, when that is confluence, when that term confluence is nothing but the when stem cells or cells occupied more than 80% of the culture, that confluence. So it is confluence when the cell all cells are occupied, the T25 plus, you just EDT tips in 2 to 3 ml. You just put it in the flask, okay, and just mechanical vibrations or put 5 minutes into the CO2 incubator. Then again, you will see due to the tipsin EDT, it lifted the stem cell which is attached in the plastic body. After giving EDT, they lift the stem cell from the plastics. So after lifting, you again take it in a test tube. Then you put again culture media. Then you centrifuge 2000 RPM for 20 minutes. You will be find just below in the glass tube, that is pellet. This pellet you just collect, wash with DPBS 2 to 3 times. Then again you culture. Again more plus, that is T75 plus. So first you go for T25 plus. When it's confluence, you go for then you for T75 plus. So passage 2. When passage 2 also confluence, you go for passage 3. So I think in passage 3, you will be find more millions of stem cells. Okay, sir. Next slide, please. Next slide. Okay. Now you can stain. You can stain, say for in the left side, that is rabbit MSC, by crystal violet staining, you can see the cytoplasmic and nucleus. And also horse ATMSC. In the right side, there's horse adipose tissue MSC by Jim staining. You can see the nucleus and the cytoplasm. This is nothing but the spindle shaped stem cells. Okay, sir. Next slide, please. Okay. Next slide. I told you as the properties of the stem cells. First property, it must be plasticarian. That I show you. Next property, it must express some positive as well as the negative marker. Positive marker like CD45, CD73, CD90, CD105, and also it must not express CD negative also, CD14, CD34, 45. By this marker study, you can do either by facts or by the next slide, please. So by the facts, you can characterizations of MSC. In the left side, you just see it is CD105 positive, and left side that is CD34 here, CD34 that is negative expressions. Next slide, please. Also, by not only by facts, by the PCR technique, you also can show negative and positive expression. Almost left this one, you see CD34 and CD45. There is no base pair you sign, so it is negative expression. Whereas this one, CD105, you just see there is a base pair, 43 base pair, CD105, 342 base pairs. Okay, and this year CD73 positive. So you can already told the second criteria as per the stem cell society, it should be some positive and negative expression cell markers. Okay. Next slide, please. Okay, just I will show you how is the stem cells in the scaling electron microscope. Okay, say for you could go this one 6 day. You just go 6 days, you can see there is a nucleus and both the side there is a cytoplasmic process. Okay, scanning electron microscope, you can see this type of stem cells. Okay, next slide, please. Next slide. Okay, now after collecting stem cells, you can produce bone cells, you can produce fat cell, you can produce cartilage cells in your lab. Okay, you take 2 flasks. 1 flask as it is control, you not added anything. In another flask, you added some, say for osteogenesis, you just added some osteogenic precursor materials. Okay, differentiate, it is marketed available, osteogenic differentiated media. Okay, after few days, you will see this cell's shape is changed. You just said this is control, the same, there is no shape change. But here, the shape is changed, cell shape change. When you go for this one, next slide, please. You can, next slide, you can show this one, above one, that is, these cells become osteoplastic cells or bone cells. Only bone cells, they stain by the alizanin red. Okay, so bone cells nucleus take the alizanin red, you can lot of bone cells develop in your lab. Second, also say adipogenic or fat cell differentiation, you can stain with oil red O, you can say the fat cells. Next slide, please. Next slide. So you can also show the chondroblast or cartilage cells. That is Alcian blue. You say the blue, Alcian blue, that is cartilage cells. Also, you can develop in your lab liver cells. Okay, by the process stem cells, you just added the differentiating media, so you can produce a cyst. Okay, next slide. So next slide. So I just gave you osteogenic differences from MSC. You can culture, you can produce bone cells in your lab and then either stain alizanin red or alkaline phosphatase, this type of picture. You get a lot of bone cells develop in your lab. Okay, next slide, please. Next slide. Next slide. Okay, this is mesenchymal stem cells with DAPI stains. Okay, this is the big form or large sizes, either DAPI stains. Next slide, please. Next slide. Okay, this is another DAPI stains. I change the filter. This is actually the figure of a stem cells. Okay, next slide. Now, next slide. So in IFRI, we can produce, we have also stem cell bank. Actually, we collected different type of stem cell from canine, mice, rabbit, guinea pig, equine, and rabbit, and we kept in liquid nitrogen or cryopreservation. We produce this stem cell banking. So whenever needed, different particular species of stem cell, we directly taking from cryopreservation, thawing it, and then directly use. So this is develop of a stem cell bank. Next slide, please. Next slide. So introductory class, what is stem cells, what is their property, how you collect, how you culture, how you differentiate, what is the properties, I just try to tell you. Now I going for their experimental produces, see whether these stem cells are able help in different type of our medical utility. First, I will go three type of study I will show you in the bone defect or fractures. Okay. In this slide, in this experiment, I cut 15 mm defect on New Zealand white rabbit in the radius. Okay. CSD means critical size defect. What is critical size defect? That defect which never heal on its own. That is called critical size defect. Okay. So here critical size defect in radius of New Zealand 15 mm. So you just see in the second, we just cut the 15 mm bone and then put in this one, I put some biological construct, okay, that is called bioceramic construct. This is bioceramic and bioadaptable and bioabsorbable bioceramic abstracts. Okay, I just put that fracture gap and then I suture it. Here in this group, we just there is 8 group. In there is group A, that is autogenous, means same animal. Group B, allogenic, means not same animal, same species but different animal. Group C, allogenic MSC plus one osteoinducer, that is called bone morphogenic protein. Group D, allogenic MSC with another osteoinducer interleukin growth factor, ILGA. And group E, that is geno, different type of species, sheep. And group F, different type of species, geno, dog. And group A, only I use the bioceramic, no stem cells. And group H or last, there is control. I just make the defect, nothing, no stem cell, no bioceramics. Okay, next slide, please. I'll hurriedly go. Next slide. Okay, we just see the first group. First group in the just you see the day 0 and day 90. In day 90, you just see the bioceramics. After stem cells, autogenous stem cells, there is a new bone developed between the two defect area as well as the adjacent ulna. Next slide, please. That is allogenic MSC. Also, you can see the ceramic is intactly attached with the defect sites and also bone developed in the new adjacent ulna. Next slide, please. Here we can give some bone morphogenic, that MSC plus we give some osteoinducer bone morphogenic protein 2. Here you find a lot of new bone develop in the proximal end of fracture, distal end of the fracture, and the surrounding ulna. Next slide, please. Here also another osteoinducer, that is interleukin growth factors. Here also new bone develop and closely, intimately attach this bioceramics with the fracture site. Next slide, please. Here you can see the most important, the experiment in the rabbit, but we use sheep MSC. We use sheep or geno MSC. That is no rejection. Rather than you can find new bone develop both the proximal end, distal end, as well as the surrounding radius. Next slide, please. You can see here we use dog, canine MSC in the rabbit experiment. Here also there is no rejection, but new bone develop both the proximal end, distal end, and the surrounding. Okay, next slide, please. Here only bioceramic. You just see only bioceramic, no MSC, or either auto, allo, or geno. You just see the bioceramic just loosely attached, no new bone developed, nothing new bone developed. Next slide, please. Next slide. Here is the control. I have given no bioceramic, no stem cell. So the fracture at day 0, day 90, as it is same, no healing. Okay. So in a nutshell of this small experiment, I can prove that stem cells, whether it's auto, allo, or geno from other species, help in fracture healing. Okay. Next slide, please. Next slide. Okay. Then, actually, this is this project. It is from DBT projects. DBT told me, Professor Maiti, why you just remove 80% of the bone? Because when there is a accident, say for accident, lot of our bone is damaged. So they told me you you remove 80% of the bone of a single bone. Then you find whether stem cells have the ability to produce newborn or not. Okay. So here instead of 15 mm, I mean just double the defect size. Okay. And you just said 3 mm, 3 centimeter, that is 30 mm, and I produces in the defect side, there is a bioceramics. Okay, next slide, please. Next slide. Okay. There are 4 groups. Okay. Next slide. Next slide. We just see. Here in group 1, you just see big defect, 30 mm, 85% bone I have cut it. So here we use the autogenous, means same individual, same animal. Here you find beautiful newborn developed. The bioceramic is closely attached, intimately attached with the surrounding ulna. There is no loosening, rather than in the proximal defect and the distal defect, there is closely attached with the newborn. Next slide, please. Here, here you can see. Here we give the, you just see in this animal, you cannot differentiate where we make defect. As it is new, whole the construct become newborn. Whole the construct, 30 mm construct become newborn. They already started remodeling. Periosteal proliferation started, medullary cavity developed, and as it is remodeled, as it is just like day 7 or day 0 bone. So a lot of new bone develop when we cut 80% of the bone. Okay, next slide. Here we use, next slide. Here we use, okay, it is that is without stem cell, only construct. We just see the, you just see the construct disintegrated, no newborn developed, because here we give in no MSC, only construct. So there is no newborn developed. Next slide, please, sir. Next slide. Here you can see. Here you can see only no ceramic, no stem cells, as it is. The bone defect remain as it is at the day 0. Okay, at the day 0. Also, I've seen here the animals, as because the long defect, all the animals are succumbed on day 45 because their lameness, no newborn developed. So day 45, maximum more or less, they are died. So in this experiment, I concluded the stem cells with alone or with osteoinducer help in newborn develop even in a big fracture defects. Okay, next slide, please, sir. Okay, this is next slide. Next slide. You just see here. Actually, this is the ulna. This is the, and this portion I cut from this to this. So this is the newborn develop. So this portion you can see newborn develop. This is the old bone. This is the old bone, and this is the newborn. And excess ceramics they get out of the newborn develop. Okay, here you can see up to this newborn develop and that size of the old bone. Next slide. This is 2-dimensional CT study. Next slide, please. And here you can see. Here you can see. Here you can see this one. This is the cut end. So new bone develop, and this is called periosteal bridging. We just see this is the periosteal bridging. This is the old bone. This is the old bone. So this is the periosteal bridging, and mediastal and excess ceramics they go outside. Okay. So this is 3D CT study. Next slide. Next slide, please. So next slide. So this is another called oxytocin level study. Oxytocin level study. Only those you are the old bone. This is the old bone that is called sea green color. Okay. But when there is new bone, there is golden yellow color. Here you see lot of new bone develop. Last osteocycle lacunae, canonically not a new bone developed. Okay, golden yellow color. That is OTC label study. Next slide, please, sir. Next slide. Next slide. This is histopathology. You can see. Make it bridge. Say for this year, this one. This is the curtain. This is the curtain. So new periosteum. New periosteum. Yeah, you can see. This is bone defect junction. Here we cut. This is NP mean new periosteum. This is old bone. This is the new bone developed. This is histopathology, and this is the scanning electron microscope. Next slide, please. Next slide, please. So in another experiment, actually this experiment I have done in Germany, Leipzig University. Actually, there they said me, Professor Maiti, you make test in one animal, control in another animal. You make a model where test as well as the control study in same animal, because he said that each animal, each human being or animal have different type of health status. So these health status, they may give enormous results. So they said you make a model where you can study the control as well as the test. So in this model, I peg a big size pigs, about 300 kilo pigs. I make in the frontal bone, I make 4 hole, okay, by bone drill, that is 4 identical, that is 20 mm and 3 mm depth, 4 hole, and I put control as well as the construct as well as the stem cells and BMP. Okay, next slide, please. You can be clarify. Next slide, please. Okay. This is my experiment. Next slide, please. Next slide. This is my experiment. Okay. I just making hole in the frontal bone. Next slide. Next slide. You just see the frontal bone I about to make drill. Next slide. Okay. I just give the skin reducer and elevated the beneath the skins, and also I scrapped all the periosteum. Actually, bone covering is the periosteum, and periosteum has the two layer. Outer is fibrous layer, inner is osteogenic or cambium layer. This inner layer also produces bone. So I have to strip the whole periosteum, otherwise they give the some enormous results. So after stripping the periostems, I clean the frontal bone. Next slide, please. Next slide. You just see I started bone equidistance, equisize 4 bone drill in the frontal bone of a big pig. Next slide, please, sir. Next slide. You just see I make 4 equisize 20 mm by 3 mm equisize for 4 defect. Okay, but it is just advisory. Here I just 3 mm depth. If you make more than 4 mm depth with the bone drill, your brain will be come out and it will be die immediately. So you have to take care. You have to drill only bone, and beneath the bone, if you drill, your brain will be come out. Okay. So this 4 drill. Next slide. 4 hole we prepare. Next slide. You just see. Next. I make one control. In this control, I not giving any material. Here I give some bioceramic, only bioceramic. Here bioceramic with stem cells. Here bioceramic, stem cells plus BMP2. Okay. So actually, this is one animal. In another animal, it is reverse. Maybe there are stem cells. In another, maybe there here is stem cell. Maybe control is there. So in a blind study, surgeons should not know in which hole, in which study they are doing. In this study, in this model, I can assess where is the only control as well as the stem cells I also agree. So in the same animal, same health status, nothing will be enormous results. So I put it different hole in different animals, different type of material. Next slide. After that, next slide. After that, I put tungsten plate so that from outer fibrous tissue or outer any tissue not invade in the our material within the bone drill. Next slide. First. Next. You just second. Fill up. Next. 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 Next, sir. Next. Next. You just see. I put tungsten so that no tissue from the outside invade in our experimental site. Okay. After that, it is some tedious job. It just takes some time. Okay. After that, I suture it. You just see. Next slide. Next slide, sir. You just see. I suture the surgical area. Next slide, please, sir. Next slide. This is my experimental animal, 18 animal experimental animal on day 90. Next slide, please. Next slide. On day 90, when I euthanize these animals and I take out the frontal bone and do histopathology, where I put only MSC, there is a beautiful bone develop. And it is very difficult to isolate. I needed chisel, hammer, everything. It is very difficult. So hard bone developed. Only MSC. And when MSC plus BMP2, lot of new bone, you can be appreciated. Okay. So this is another study of bone modeling. Next slide, please, sir. I also uses some nanomaterial. Okay. For example, in 6 group, next slide, please. Next slide. In the 6 group, I create a fracture in 15 mm. In group 1, I give nothing, only bamboo spleen. And second group, PCL plus nanohydroxide. Third group, PCL nanohydroxide plus microwall carbon nanotube. In fourth group, PCL nanohydroxyapatite, then carbon nanotube plus BMP2, osteoinducer. Fifth group, everything, instead of BMP2, I get RAD MSC. Okay. And last group, I've got all the MSC plus BMP2. Okay, next slide. Next slide. You just see the above one is PCL plus nanohydroxyapatite, and this is carbon nanotube. Okay, next slide. You just see, next slide. Okay, I put 15 mm critical size defect, and I put the PCL bioscaffold. Okay, next slide. Okay, you can see. After I put there. Next slide. Okay, you just see. Next slide. That is carbon nanotube. I put here. This one. I put in the fracture line, and after sutured the peristome and skin. Okay, next slide. Okay. Sir, in this goal, you just see. Next slide. Next slide, sir. Next, you just see in the group A. You just इसका थोड़ा छोटा करिए, sir. You just next slide. Next. छोटा, हाँ, और छोटा. You just see in the day 60. You just see. No, in the first group, group A. You just see group A. Nothing, as because we have nothing. No bioceramic, no, the fracture lines as it is. No bridging callus is developed. Next slide, please, sir. B. Only you just PCL and hydroxyapatite, slightly new bone developed in the both the cartilage. Next slide, please. In group C, the carbon nanotube used better than nanohydroxyapatite, more new bone developed within the cartilage. Next slide, please. Next slide. Group D, where is carbon nanotube plus BMP2, lot of new bone developed, but still there is fracture. Fracture is not completely obliterated. Next slide, please. Group E, we just see when there is RMSC is given, lot of new bone developed, also bridging callus developed. Next slide, please. Next slide. And that is the last group where we use stem cells as well as the growth protein. You can see only day 30 you just see almost 70% healing, and day 60 fully new bone develop. You cannot differentiate where we cut it, and where we use the bioceramics. Whole bone is new bone developed. Okay, thank you, sir. Next slide. Next experiment. I go hurriedly. Next element, sir. Just go behind, back. Next. In this, I make heel in the rat model, 2 into 2 centimeter. I cut the skin on model. In group one, I just give after 2 into 2 centimeter skin wound, I give the only dermapine. Second is chitogen, medical chitogen. Third is giving, in the MSC. And fourth, MSC plus chitogen. Okay, next slide. You just see. Next slide. You just see. This one, group 2. You just see in the group. This one. इसका आगे चलिए सर आगे ग्रुप 3 ग्रुप ना आगे बैक this next sir you just see in the group 4 नहीं आ रहा है group 4 इसके बाद थोड़ा सर साइड्स में सर थोड़ा ग्रुप 3 देखो ना उससे पहले ग्रुप 3 का थोड़ा छोटा करिए ना जाइए थोड़ा छोटा करिए अच्छा चलो देखना ग्रुप 2 आ रहा है ना ग्रुप 3 इसके बाद ग्रुप 4 भी है सर साइड में देखिए ग्रुप 4 वहां लिखा है बेस्ट ग्रुप ओके ग्रुप यहाँ देखिए
दिख रहा है सर ग्रुप 4 है ग्रुप 4 इज देयर सर
but here is not seen my okay you just see group 4 you just see so first is 0 day 7th day 14th day 21st day and 28th you just see when we use in the skin defect both the chitogen and stem cells new hair is developed and not able to differentiate where we made the skin defect but in the first you just see we give nothing as it is present. So, in a nutshell, the skin defect when you use stem cells with chitogen, new bone and new hair is developed as it is normal. Okay, next slide, please. This is the next slide. This is the histopathology. You just see, ये छोटा करे group 4 नहीं आ रहा है अच्छा आ रहा है group 4 आ रहा है you just see in group 4 this is the best group you shall see that is epithelialization started, hair follicles also started by the HNE stem and mesenteric stems. So, in general, MSC potentiate skin wound healing and this MSC is immune potential. We take MSC from other species, here with rat, we use, it is immune privileged. No immunogenic reactions. Next experiment. Next. Oh, we just small it. Okay, sir. You just see in the first slide, that is the scanning electron, lots of stem cells you can find there. Lots of stem cells you can find. This is lots of stem cells. This is lots of stem cells you can find. Okay, sir. Next slide. Next experiment. I make corneal defect. Okay. I mean next you can go. Next. I make corneal defect in the rabbit. Okay. I make cornea. Rabbit I make defining the cornea and I use their wound and evaluated by the fluorescent, rose bengal and Leishmanian Gonstein. Okay. And next slide. You just see. So in 4 groups, in first group, after defining the eye cornea, I use decellularized cornea. Okay. Decellularized means after decellularizing, that indigeneity is lost. This is porcine origin. I use porcine decellularized cornea. Here I use porcine decellularized small intestine submucosa. Okay. And here cornea plus stem cells, SIS, small intestine plus stem cells. Okay. Next slide. Okay. You can see there is 6 group. You just see group C. This slide. This is the beautiful results and after ophthalmic dye or fluorescent dye or West Bengal dye, you can, they're almost same vision is coming after using stem cells. Okay, is them, and along with the bioscaffold we use here in the decellularized cornea. Okay, decellularized porcine cornea. In the rabbit, we use decellularized porcine cornea plus stem cell, and in this group, as well as the eye appearance as it is normal. Okay, so wound repair with RMSC seeded acellular porcine cornea, it help in corneal wound defect. Next slide, please, sir. Next slide. Okay. Next, I will show you the burn wound. Burn wound. You know, there is first degree burn, third degree burn, fourth degree burn. Okay. In fourth degree burn, I produces in rat model fourth degree burn, full thickness, full skin burn, and in group A, I just giving silver X, where in human also is Bernal or silver X. Second group we give porcine acellular urinary bladder, C porcine urinary bladder plus I make a gene code that is pDNA PDJ beta. D group with stem cells, and last group that is PDJ beta along with the stem cells. Next slide, please. Just next slide. Next slide. You just see this one. You just see this one. This group where I put stem cells as the PDJ beta. You just see nothing. You can be differentiate. Where is the burn wound? Here is the burn wound. Burn wound. Okay, day 7, day 14, and day 28 in group 4. Okay, you see there is a beautiful all hair develop. All scar tissue, burn scar tissue is vanished, and new hair is developed. So, in a nutshell, that is stem cells and PDJ beta which you produce. It is help in the burn wound healing. Next slide, please, sir. Next slide. Next slide. Okay. Now I'll get another. Okay, I'll go. Just I'll go. Okay, next slide. You just go right. Next slide. Next slide. Next slide. Next slide. Yeah. No. It's in this model. In this model. Okay. Actually, there are in the rat these yellow portions I resected. About 65 to 70% of liver I resected, cut out. Only 15 to 20% these portions of liver I kept. This portion I liver cut out, I kept. Then I use stem cells. Whether the new liver is produced or not. Okay. Next slide. You just see. After using anesthesia, this surgical and these portions of the 70 to 75% of the liver low, I resected. Only 15 to 20% of liver kept it inside, and then I suture it. Okay. Next slide, please. You just see. This is. So these portions of liver I cut it, and after 15 to 20%, only remaining liver I put inside, and then I suture with the Vicryl 30. Okay. Next slide. I just go to the results. Next, you just in the control group. Where I am not giving anything. Okay. You just see day 20 liver size, day 40 liver size. You just go, just see. Next slide. This is control group. Next slide. This is stem cell group. You just see. The best group, this one, stem cell plus epidermal growth factor. You can see this is the liver where it is. After giving stem cells, their hyperplasia means new liver cells is developed. As it is the normal size before cut it. Okay. Next slide. I'll show you. Next slide. Okay. Actually, sir, in the control animal, we see where we not giving stem cells, there is cirrhosis developed, fibrosis developed, collagen fiber is developed within the liver. Okay. Next slide. Next slide. And this I go for next I go for histopathology. Next उसका पहले सर पहले उसका पहले we just see in the control animal where we not give any stem cells, the liver is also a very small size, Their fibrous developed grossly, and histopathology, lot of fibrous tissue developed. Next slide. Next slide. You just see. After euthanasia, actually this is, you can see, this is the cart area. This is the cart area. So these portions of liver, new liver hepatogenesis. Okay, one thing. And you can see the color, the texture, the appearance of the liver as it is normal. But in these animals, we cut it 80% liver, we remove it. But after stem cells, after day 40, when you euthanize the animal, when you remove, when you open the abdomen, we can see their liver appearance, color, their texture, their juicy appearance, as it is normal liver, not regenerate liver. Here you can see this is the regenerate person, this is the cart area, you look, cart area. So these portions are regenerate. Okay, next slide, please. Next slide. Here you can see this is the cart area, these portions of the liver newly developed. Next slide. So we go for histopathology. This is gross histopathology. You can see all the liver triad, that is portal vein, hepatic artery, and liver cell. Liver triad is developed. This is the regenerated liver cells. Okay. Here we removed 80% liver. This study I done when our liver is damaged. When our liver is damaged, either say for bacterial culture or some plant origin or something, or by alcohol or something, liver damage, only therapy is for liver transplantation. But liver transplantation is a very huge amount of cost. Also, the survivability is sometimes not of contention. But in this study, I show you when I removed 80% liver, I put stem cells, new liver cells, and as it is normal liver structure, color, and histopathologically the same liver. Okay, sir. Next time. Next. Another experiment I will show you. Next. Yes. Cirrhosis of liver is a great concern of human. Okay. Cirrhosis. Oh, particularly those who are drunkard or something. Cirrhosis liver. So here I made a you just see there is a bile duct. This is the bile duct. This is the liver. This is the duodenum. This is the bile duct. Okay. I ligated bile duct here. Here. I ligated above and below, then cut it. So bile formation as it is normal. But that bile, due to cut bile duct, it does not go to the duodenum or the feces. So more and more accumulation of liver within the bile, within the liver. So I make induced cirrhosis by accumulation of the liver. Next slide, please. Next slide. You can see. After 5 days of liver ligation, the color of liver all become icterous. Not only, also the surrounding duodenum, surrounding intestines, they are also become icterous. That is, I induced cirrhosis of liver. Okay, next slide. And here, in these cases, I also go for histopathology, lot of necrosis of liver. Next slide, please, sir. Next slide. You can see, after 40 days of post-ligation of bile duct, liver, this color, texture, okay, and color within the body, outside of the body, and here is the histopathology, just like normal liver. It is not showing it is icterous liver or cirrhosis liver. Okay, sir. Actually, I go through the journal. I have seen this actually in our liver, our called cirrhosis or liver damage. The collagen fiber deposited in the liver becomes cirrhosis. In the liver, there is a called satellite cell. Other than liver cell, there is a satellite cell. These stem cells, if I go to the first or बिल्कुल पहले वाला slide में आप देखोगे, these stem cells, they three procedures. Either they kill the satellite cell, either the proliferation is reduced, either they reduce the collagen fibers. So on the process by three procedures, they reduces the depositions of collagens in the cirrhotic livers. So you can find here In the normal animal, I show where I am not giving any stem cell, what is the color, texture is different. But after giving this texture, you can find the liver as well as histopathology, just like normals. Okay, sir. Next slide, please. Next slide, I just go you. Next slide, I just go. Okay. Then I show that another very important, it is called nerve damage, paralysis. The nerve only cells when it's damaged or dead, never, very slow regenerations. That's why paralysis comes. Here I use some biomaterials or nanoscaffold after I damaged the sciatic nerves. Okay, next slide. Next slide, please, sir. Next slide. You just see. You just see. Here, I take the sciatic nerve outside and then twist it. I twist it so that I completely damage this sciatic nerve. Completely damage this sciatic nerve. This sciatic nerve supply the gastrocnemius muscle. We just see I completely paralysis the rat because the sciatic nerve is damaged, supply in the gastrocnemius muscle damaged. So dragged. They started dragging. But here I just put on the left side, right side is normal, so that I don't want to kill immediately. Okay, but you can see they're dragging in the soil. Okay, next, because our model was paralysis, so I completed that paralysis. Next slide, please, sir. Next slide. Next slide. This is my experimental element. Next slide. Next slide. I just show. This is last slide. Okay, you can see this is the control element as it is normal. As it is normal after my experiment, even it is dragging. But where I give the stem cells in the sciatic nerve injury, you can see they started jumping. They're putting full weight and they're jumping. But here, where I'm not giving anything, only injury and give some micro scaffold. This type of scaffold I used. Just wrap on the sciatic nerve, injured sciatic nerve, wrapped, and then I use stem cells. You just see the animal started. I also video the animal can jumping, running. Okay, okay. Thank you, sir. Next slide. Next, you can see. Next, just piche. Yes, you can see the scanning electron microscopy. Here is the nerve. You can show it is injured. It is injured. Okay, it is disconnect. But here you can see it is regenerate. After giving stem cells, it is regenerated. Here the gastrocnemius muscle, this is atrophy of the gastrocnemius muscle. Here after stem cells, regain of the gastrocnemius as it is normal. So in a nutshell, stem cells helps in nerve regeneration. Stem cell helps in atrophy of the paralysis muscles. Again, it regain the muscles. Okay, next slide. Next slide. Cartilage. You know, just like nerve, cartilage tissue also very slowly it is healing. So after cartilage damage, I put this type of nanoscaffold. Okay, this type of nanoscaffold I put. Next slide, please, sir. Next slide. This is this nanoscaffold attached with the stem cells. Okay, you can see the depistem, lot of stem cells attached within the nanoscaffold. Okay, next slide. I just I just go next. Okay, you just see here the staple joint. Okay, I open the staple joint and I put here cartilage drill damage. Okay, here you can cartilage damage. After cartilage damage, with this scaffold and stem cells, I fill up this damaged cartilage. Okay, and this is the X-ray. You can see this is the X-ray. X-ray after staple joint drill, I fill up with this this type of two type of scaffold I use along with the stem cell. Okay, next slide. So next slide. Yeah. Okay. This is we just said day 60. This is where I nothing put anything. Here you just put stem cells plus scaffold, the cartilage healed and calyceus is open. So next slide. Next, on day 90, you just see there are glistening appearance, completely healed cartilage. Here we make the drill. When after put the scaffold, alloys with the stem cells, he just completely healed. This is only stem cell, this is stem cells with the growth protein. Completely healed, the glistening appearance. Okay. So last two experiment, my aim was those cells are very late regenerations, particularly nerve and cartilage. I tried to see whether stem cells has the ability can produce nerve cells or cartilage cells. So I can show you. Next slide, please, sir. So next slide. So this is the staining proof that is cartilage cells. New cartilage cells are developed. New cartilage cells are developed. Histopathology. Next, this is another that is SNGP plus BMP with a lot of cartilage cells developed. Different type of stains, lot of cartilage cells developed. Okay, I can show you. Next, you can see this is the scanning electron microscope. Lot of new chondroblast is developed in the defect sites. But here is the contour, very less. This is the contour. Okay. Another only scaffold, nothing in MSC, nothing is developed. Here you can see BMMSC plus growth protein 5. Okay, lot of cartilage cell. It is called chondroblastic mounds. Okay, you can see. Okay, sir. Next. I just okay, wrap up my deliberation. This in Cologne University, this is stem cells hospital. I have there. This is stem cells, the all type of human fracture, starting from fractures or liver disease or paralysis or HIV or Parkinson disease. They only use the stem cells in this hospital. This is the cell center, cell hospitals. Okay. Next slide, please, sir. Next. Nowadays, another new generation of stem cell called iPS cell, induced pluripotent stem cells. Here, no need of bone marrow. Even you can take a small skin tissue, small, 1 to 2 millimeter, and we change the genetic change of the skin cells. You can derive either nerve cells or the heart cells. Okay, you can different type of cells from the skin cells, just genetic coding change. Okay, that is new technology is called iPS cells. Next, I'll just show you. Okay, sir. Double click. No. We go to the end show. It is not the full slide. You go there in the just only slide. Okay. Open, sir. It is not open, sir. Double click media player.
It's open, sir,
I am not able to see.
but it's running now.
But I have not seen, sir. It is running.
अच्छा, it's just a minute, but it's
You can show in your lab, you can produce your own heart cell. That is my intention. Sir, you go not the full view of slide. You go to the some small slide slow, and then you open, double click, open it, open.
now it's showing.
मेरे पास नहीं आ रहा. I do not know.
नहीं आ
आप
रहा.
देख रहे हैं?
हाँ, मुझे दिख रहा है.
Sir,
आपको
it's
दिख रहा है?
under process. It's under process. It's being
Huh?
loading.
Loading.
Yes, sir, I think it's being loading
ये
because
तो ये मतलब दिख
yes,
रहा
sir,
है,
it's
इस पे लिखा आ रहा है योर स्क्रीन इज शेयरिंग।
सर अभी शो स्लाइड शो हो रही है पर वीडियो शो नहीं होना शुरू हुई।
वीडियो
जो ये वीडियो का वो बना हुआ है आइकन, एक बार आप उस पर क्लिक करेंगे।
ये देखो अब कुछ हुआ।
नहीं, जहाँ सर वो बिल्कुल सेंटर में है।
एक मिनट देखो, ये अभी चेंज हुआ ना।
हाँ जी सर, स्लाइड चेंज हुई।
And next.
अभी चेंज हो गया, ठीक है। अब
अब
ये
यहाँ पे
वीडियो
बिल्कुल
पे क्लिक किया
नहीं,
मैंने।
बॉक्स पे नहीं सर, बॉक्स पे नहीं, बॉक्स की जगह सेंटर में।
इधर जो साइड है, इधर जो साइड है, नॉट फुल व्यू।
हाँ, फुल व्यू नहीं
ओनली,
है सर ये।
अच्छा फुल भी नहीं है तो करो, ओपन करो, ओपन, ओपन।
अब आया।
नो, इट इज नॉट सही।
अब इधर तो आ गया।
यू कैन शो द
हाँ,
हार्ट।
सर आप अपनी स्क्रीन शेयर कर दीजिए अगर आपकी तो
अच्छा
शायद
अच्छा अच्छा,
स्टॉप करके।
मैं करता
ओके,
हूँ
मेरे
सारी
पास
स्लाइड आ रहा है यहाँ पे मतलब।
but,
एक मिनट
but
कुछ करते हैं।
all you are showing that is heart cell contractile cardiomyocyte. It is contractile my beating my cardiocyte.
अभी करता
Can
हूँ,
you
मैं
see?
करता हूँ जस्ट। अब देखिए सर।
एक मिनट, let it come. Yes, yes. Now it is the, I just show you. अभी देखो, you can at small skin, then you produce your own heart cell in your lab. Show if there is air, ये तो आगे नहीं चल रहा है, कॉन्ट्रैक्टाइल नहीं हो रहा है।
होगा सर
Here
अभी एक।
हाँ हाँ, अभी आ रहा है, तो देखो, you just see your heart cell in your lab. You can produce by the IPS technology. You just take a small skin cells and genetic coding so that you can produce your heart cell in your lab so that if any emergency is a heart attack, you can directly put your own heart cell. Okay. Okay. You can see. Next. This is totally next. देखिए,
ओके सर।
I
हाँ,
just next single heart cell I will show. Single cardiomyocyte. This is as a flask. This is this is called beating cardiomyocyte, beating heart cells. In the lab, you can produce. Next cell, next
नेक्स्ट वीडियो।
one.
हाँ,
उसके और एक लास्ट के जस्ट पहले, लास्ट के बिल्कुल पहले। Okay, I think it is not possible. Okay, sir.
सर करता हूँ, मैं करता हूँ, बस हो गया जस्ट। भी
Okay,
अपने ऑन
great.
किया है। एक्चुअली अलग से करना पड़ रहा है विदाउट स्लाइड।
Okay. हाँ,
ये आ
you
गया।
just see. This is the single heart cell. ये कॉन्ट्रैक्टाइल नहीं हो रहा है, वो कॉन्ट्रैक्टाइल होता है।
होगा सर, अभी होगा।
You
ये
just
देखिए।
see. Yes, this is the single heart cell. So my intention was, by this technology, you can produce your heart cell in your lab. Okay, sir? Okay, this is called beating my single beating heart cells. Okay, sir. Thank you. Last slide, sir. Thank you,
थैंक यू
sir.
सर, नाइस प्रेजेंटेशन,
Last slide.
वेरी इंफॉर्मेटिव दिस वन। जस्ट अ
Very
मिनट।
last slide. So,
नाउ आई विल शेयर अगेन दैट स्लाइड्स।
yeah, this is the thank you slide. Actually, this is my best place. I'm in my world tour. I know This is Holland tulip flower, tulip garden. Whole field is tulip, full of tulip. Okay, thank you all. And I have one book. Book name is Stem Cell Research in Lab Animals by Swapan Kumar Maiti. You just go to the Google and all everything technology, the preparations, everything it is nicely depicted on that book. Stem Cell Research in Lab Animals by Swapan Kumar Maiti. It is available in Amazon. Amazon. Okay, but the publishers they give some right. It is about 4,000 per book. It is available. Whatever my slides, whatever technology, how you culture, how you differentiate, everything is published in my book, Stem Cell Research in Lab Elements by Swapan Kumar Maiti. Okay, sir. Thank you, sir. Thank you,
Thank you
everybody.
so much, sir.
You
Thank you so
all.
much, sir, for enlightening us.
Okay,
So
sir.
now the session is open for question and answer. Participant may post their questions in chat box.
Okay, sir.
Participants are requested to kindly post their questions, their queries in chat box. So if there is no query, then
Okay.
the session we are closing session. So thank you so much, sir, for such creative session and enlightening the participants with your views, with your experience. Thank you so much, sir.
Yes, sir. Okay. Thank you. Thank you. Thank you. Thank you, sir.
Moving forward to the technical session two. So now I would like to introduce our second speaker, Dr. T. Giraneshwari Devi. T. G. Devi is Assistant Professor in the Department of Livestock Production Management at the College of Veterinary Science and Animal Husbandry, CAU, Jalukie, Nagaland. She holds a PhD in Livestock Production Management and has qualified NET in Animal Production with over 15 years of experience. She has served in various roles, including veterinary surgeon, research fellow, and assistant professor in different institutes across India. She has received recognition of excellence in her field and has contributed significantly to 18 research paper, books, manual, and extension publication. She has been actively involved in teaching, research, extension activities, including farmer training, project implementation, and capacity building programs. So now I would like to request Dr. T.G. Devi to kindly deliver her lecture on for technical session 2.
Dr. Nisha, I think one question is there for sir. You can check that one. Possible.
So, sir, there's one query. Someone is asking, Nikita is asking, in a nature nerve injury, we want to know exact spot of the injury. How can we apply MSC in such cases?
I just on the site of injury. I inject stem cell in the site of injury.
Okay, sir.
Okay, okay.
Any other query? So, thank you so much, sir. So, now I would like to invite Dr. Gyaneshwari Devi to kindly deliver her lecture note.
Uh, Good evening, everyone. Thank you very much, ma'am. Today my lecture is on Bokasi pig farming, which is an innovative, eco-friendly technology, and also it has a vast opportunity and challenges. So to begin with, I would like to just introduce our notice, which is, as you all know, that means among all the livestock, pig is considered as one of the most important livestock by the farmers. So, like after observing how they are rearing, so what are the problem they are facing? like especially smell to the locality, and also like means problem with the neighbors. And also considering all this problem, I took up my research work on Bokasi pig farming. Next slide. Yes. So Bokasi, it is a Japanese technology which is around 10 years ago. So next slide. Yeah, Bokasi, it means fermented organic material, which is also known as the IMO, that is indigenous microorganism, which help in reducing the odor, that is a smell, which is means you can find in the pig set. And also it prevent flies, and it improves the when fed this IMO in the feed, it increase the digestive system, and also it promote healthy growth and also overall management of the farm. Next slide. So the means Bokasi pig farming, it is usually the design of the housing. The design, it is very much different from the conventional and then traditional system, which usually normally we are practicing. So here, first of all, the site selection is very important. So first we have to select those sites which are means elevated area or upland area where waterlogging is free, and also the orientation as it is like east to west direction. And the most important is there should be no any seepage of the water or any water nearby water resources. This is the main thing, so as to prevent the water seeping into the set. Next slide. Next slide. So this is how we constructed. After selecting the site, which is upland area or elevated area, so the first thing is we have to make a tank which is of 3 feet height, and then above this tank we have to make the half wall which is also a 3 feet. So from the ridge till this means where we are making the plaster, it is altogether it is 10 feet, and from the wall to the eave, that is 8 feet. So this is the measurement what how we have to make. And this can be concrete also, or else we can always make with the locally available material like those wooden plank and all. Next slide. So here, like in this tank, we have to fill with those biomass that is around 10%. First of all, we have to choose those bedding material which is easily available and which is free of cost. So first of all, we can go for in our place sodas is available without any payment. So we choose those bedding material which is easily available. So we 40% of those sawdust, dry biomass like those leaves, and then any dry even the straw also can be taken. So 10% of it. Fresh soil means which is not contaminated at all. So those fresh soil, it is also known as the virgin soil, can be taken, that is 20%. And then charcoal should be grind properly, powdery type, and also rice husks 20%. And all these ingredients should be mixed properly by using a raker, turning upside down and all. And then after that, we have to make it moist, that is 60% moist, by sprinkling water on top of these bedding materials. Next slide. So you can see from the picture itself. Next slide. So now IMO, this is the IMO which we prepare by ourselves. So the IMO will be sprinkled on top of those bedding materials, around 4 ml in 1 liter of water. So this IMO is very important. If you don't prepare well, then the whole thing will be spoiled. So thereby IMO, that is indigenous microorganism preparation, is very important. Next slide. So after sprinkling the IMO on top of the bedding material, we'll keep it for 1 week so that those microorganisms will be activated on it. Then after 1 week, then we will those wind piglet will be will be this one introduced into the Bokasi pig unit, and then you will see those natural behavior like rooting, and then they are means means they are been adjusted to that new environment. So if the IMO is not prepared well, then the animal may try to turn away from that area. So thereby you will observe those behavior, and then thereby you can assume that if the animal is showing also all those natural behavior, you can assume that the IMO preparation is successful. And then like this here, we don't need to change the litter material every time here, and then you don't need to take out the, those fecal material and all. You don't need to remove for the till the marketing, from the weaning, introducing through the in the weaning, after weaning until the marketing, say about 6 to 7 months or till 8 months, we don't need to remove anything out there from there, neither the litter material or bedding materials, or neither the fecal material. So the animal will be kept. So you can just see that labor cost also will be reducing here, and also surrounding all the this fecal material will be accumulated in that pit, and thereby there is no much labor is needed here. And so every time we have to sprinkle those IMO prepared same concentration only, that is in 1 liter of water, 4 ml will be mixed, and then after say about in 1 week 3 times you can sprinkle and then level it properly. And also you can mix the IMO in say about 5 ml of IMO into the feed like twice in a week and all. You can always go for. So here the most important is to label the IMO which you are mixing at least 3 times in a week. So proper labeling is very necessary here. Next slide. So here regarding the feeding, those waterer and then the feeding trough and all, you have to put nearby the door, so that any spill of water will not accumulate, will not be spread in the other area. So make sure that you put those water feeding tub and then watering trough nearby the door. And then other feeding part, this as normal, like 2 times in a day, that is morning and the evening. So in the between you can always give locally available green fodders and all. Next slide. So this is about the feeding of the pig, that is like how much concentrate, how much this one digestible crude protein during the starter and all. So this is normal only. So I'm not going through this. So please, next slide. So what is the advantage of Bokasi pig farming is that, first of all, no smell at all. You won't find any smell, even though you don't remove the fecal material and then you don't clean every day, still you not find any smell until and unless you, till the time when the animal is marketed. And then since there is no smell, so no flies problem will be there. And then there will be reduce in the labor also, because they don't need to clean every day. So labor cost will be reducing, and also there is a improved digestion of the animal. And because of the provide this IMO which you are feeding into the feed, which you are including into the feed, and thereby growth and then body weight, everything is improved by using this IMO. And then this one, the fat layer also reduce under this farming system. And also we don't need to use any growth promoter or antibiotics so as to increase the body weight or increase the growth and all. Here you don't need to use all those growth promoter and antibiotic. And then animal, they are very comfortable in this environment, because they are been free. They are not been as per our conventional system, where they are been kept continuously into a confined small area. Here the animal can be freely roam, and then they show all the natural behavior also. And at the time of marketing, the best part is that at the time of marketing, around 2 to 3 tons of manure is available for sale, that is along with the animal, which may weight around 60 to 70 kg body weight, that is as per the indigenous, and as per the exotic it may be more. And then along with the this animal, you can always sell the manure also. And this Bokasi manure is very rich in, when compared with the normal this one pig manure, it is more better rich in. Next slide. So and also it is very eco-friendly type of pig farming. So pig is considered as a dirty animal, but when you rear pig under this Bokase system, you will find that no smell at all, and you will consider pig as the cleanest animal, and increase in the immunity also because of the use of the probiotic. Next slide. So what is the disadvantage of pig farming is that initial investment is high, and also like waterlogging chances, like if you don't construct in a if you construct in a lowland area, there is high chance that waterlogging will be there. And also filling up of those 3 feet depth tank to fill up with the litter material, it is quite a laborious job. And also like sometimes water, when you don't when you put the water in the middle of the this Bokasi unit, then there is a high chance that you spill out of those water. And also for that, I always suggest you to put the water and the feeding trough nearby the door. And also sometime if you don't prepare IMO well, then failure may be there, and then whole process will be gone. And then there is a laborious job again while taking out those this labeling and all, everyday labeling and all. Not that much labor, but still, like, you need to take care of this. Like every day you have to turn upside down and then label it properly for proper mixing of those IMO, and then multiplication of the IMO. And also, after 8 months, when you remove those manure, which is filled with, say, about 2 to 3 tons of manure, it is quite laborious job. Other than that, like this, I would like to suggest that this type of pig farming, that is Bokasi pig farming, is the best technology. And then it is very much suited for our northeast region. So with this, I would like to thank you very much.
Thank you so much, ma'am. Such an informative session. So participants are requested to kindly post their questions in the chat box.
Thank you, ma'am.
Thank you, sir.
Participants are again requested to kindly post their questions in the chat box. So, madam, there is one query that someone is want to know how to prepare IMO.
Yeah, this question is usually very commonly asked, but right now, like, we are means for further validation and all, we are going to approach. So detail, I don't want to say. This is made with the locally available material. So since research is still going on, and we are going to go for further, validation and all. So thereby, I don't want to exactly to say everything. Yeah. So very soon, like, I will inform very soon.
Ma'am, it is like a EM technology, Japanese technology, Dr.
Yes,
Gita.
yes, 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 yes. But under that Japanese technology, like, different type of IMO can be made. So we use with our locally material which is available in our place.
Okay. Any other query or questions?
And this IMO will be very soon available on market also.
Okay.
They are this one is I mean you can keep it for 6 months.
So thank you so much, ma'am, for enlightening us. So with
Thank you very
this,
much, ma'am.
we are concluding the day 8. So now, on behalf of organizing committee, I would like to express our sincere gratitude to all the dignitaries, participants, and our speakers. Thank you everyone for your active participation in today's technical sessions. With this, we are concluding the day 8 of 10 days international training come workshop on advances and innovation in veterinary science for viable livestock practices. We look forward to your presence tomorrow for another enriching session with our distinguished experts. Thank you once again and Jai Hind.